right, I think we can begin. Um, so just for today's session, I'm going to give a brief bio for the first sort of 5 minutes, just introducing Conor Meehan, who is then going to be presenting for about 40 minutes. And then at the end, we'll have about a 10-minute Q&A session. But for all listeners, please feel free to post your questions in the chat during the session, and we will answer them when we get the chance. So good day, everyone. My name is Keegan, and I am a bioinformatics training coordinator at FHEA. We'll be moderating today. Before we dive into today's session, I'd just like to take a moment to spotlight the Phylogenetics working group at FHEA. This group brings together individuals passionate about evolutionary biology, genomic epidemiology, and the computational methods that support these fields. It serves as a collaborative space for sharing expertise as well as fostering continuous learning. And if you believe that any of these topics align with your interests or with your professional work, we encourage you to join and contribute towards this dynamic community. Today, I'm pleased to introduce Conor Meehan, an associate professor in microbial bioinformatics at Nottingham Trent University. Conor brings over a decade of experience in microbial genomics with a focus on Mycobacterium tuberculosis, as well as pathogen-informed clinical decision-making. Conor holds a PhD in HIV drug resistance and transmission and is a fellow of the Higher Education Academy and a member of the Royal Society of Biology. Conor, would you please kindly take over? Thank you.
No problem. Thanks so much for having me today. I'm just gonna let me just share these slides here, and hopefully this will work. Hopefully you can see my slides there. We'll pop on the laser pointer. So today I'm just going to talk a little bit about a very quick overview of some phylogenetic methods. So this is kind of an intro to different ways that you can build a phylogenetic tree, and I'll use Mycobacterium tuberculosis throughout as a little bit of a case study. So a lot of what I'm going through today is kind of from a 2-week course down to a few day course down to this kind of 40 minutes. So we will be hop skipping and jumping through, but it's more to give you a flavor of which methods are appropriate in what circumstances, where we would use these when we're trying to go through different types of you see the slideshow and not the other one. Perfect. This is why I don't like. Can you see the slideshow now? Okay, so let me pop you onto my other screen so I can see what I'm doing. There we go. So yeah, so this is hopefully going to go through which methods are appropriate in what situation, when to use them, when not to use them, and in some places I'll point you to some other courses, including an online course that I have. But I'm happy to take questions, as we said at the end, and my email address is here and on the slides, which will be about if you need them. So please do email me if you need anything clarified afterwards. I'm happy to point you to more materials afterwards as well. So let's talk a little bit about Mycobacterium tuberculosis first and the type of data that we deal with in this pathogen. So MTB, or Mycobacterium tuberculosis, I'm going to call it MTB throughout because it's too long to say over and over. It's a causative agent of tuberculosis. TB, we have about 10 million cases a year, not evenly spread around the country, around the world, sorry, mainly in sub-Saharan Africa, over in the Indian subcontinent and down into Southeast Asia. About 500,000 of these would be drug-resistant cases, and transmission is through aerosols. So similar to what we saw with COVID-19 or SARS-CoV-2 and things like that, through breathing, through coughing is where we're getting it. Transmission estimations in tuberculosis and MTB are quite difficult because we have these long latency periods. So someone may become infected but not have active disease for maybe a year afterwards, and they could be transmitting through that period. That's a bit contentious. In drug resistance, transmission is also really important. So we want to know who's infecting whom, but a lot of drug resistant related cases are also due to transmission. So it's important not only from a public health aspect, but also from a clinical aspect in terms of antimicrobial resistance tracking. How you identify transmission networks in MTB is not always super straightforward and is very much an evolving area. When I started working in MTB in 2014, um, I was kind of bombarded by all these different ways that you could go about finding recent transmission. We were working on a study in the Gambia, and my first task was trying to pick the best method for us to figure out what if there was recent transmission occurring, and it's what 11 years later, and I'm still working on this area. But briefly, there are three different ways that people sometimes go about it. The first is the one with the lowest resolution, so not so great for recent transmission, but for older transmission, but is still quite used quite a lot, is what's called the spoligotype or the direct repeat region area. This is based on CRISPR patterns, if you've ever heard of CRISPRs. And what this is kind of like the immune system of a bacterial cell. And they will have specific repeated, direct repeat nucleotides within a very specific region of the genome. And we use 23 of these repeats and the presence absence of it in a strain to say whether to kind of give it a fingerprint. So if we have 23 of them here, you can see strain 1 has all of them present. Strain 2 has all of the first few present. It's missing 4 in the middle, so the nucleotides are not present there for that repeat, and then they're present in the last 4. And for strain 3, we only have the first 5 present. This kind of gives us a fingerprint for a specific strain, which is called a spoligotype. And we represent these in computers with ones and zeros, which I'll show you in a minute. The next step up in terms of resolution is what's called MIR-U VNTR, Mycobacterium interspersed repeat units variable number tandem repeats. So it's similar to the spoligotype in that it's repeated regions of DNA that are very close to each other, but they're spread around there are regions spread around the whole genome that we use, and we normally use, I think it's 22 of these spread around the genome. So in this kind of this MIR-U VNTR pattern we have here, it's got 4 copies of the direct repeat at this locus, 3 copies at this one, 2 at this one, and 5 at this one. I won't talk much about MIR-U VNTR. It's not really used much anymore. It can be expensive to do, and for the equivalent price, you can get whole genome sequencing. So whole genome sequencing is now the standard way to do recent transmission. How we do this is we take a genome, sequence the reads from it, map these back to a reference genome. For MTB, this is H37RV. And then for 2 isolates which have been mapped, we compare them to each other in terms of the SNPs, single nucleotide polymorphisms that are present or absent. And if they differ by less than 5 SNPs, then we say that they're almost definitely within a recent transmission cluster. If they differ by more than 12 SNPs, we would say that they're not in a recent transmission cluster. And it allows us to create these small clusters like this. So this is saying that this isolate is 1 SNP away from this isolate, and this isolate is 2 SNPs away from this isolate. That would mean that this isolate is 3 SNPs from this isolate. Here we have 3, so this is where we sequenced all 3 isolates. Here we have 3 different isolates that we've sequenced, and this one is 8 plus 2, 10 away from this one. This is 2 plus 2, 4 away from this one, and this is 2 plus 8, 10 away from this one. So we're all within the 12 SNP cutoff there, and we're keeping it very, very tight. So spoligotype and whole genome sequencing within SNP cutoff clusters are what I'm going to talk about mostly today. And the data can look like this. So this is our MIR-U VNTR loci, where we've got the number of the direct repeat at each of these different loci. We've got a spoligotype pattern here where you see the spoligotype, the spacer is absent in all of these and present here, present in all of these and absent here. And from this, we can create some kind of tree, and I'll talk about this in a minute. And then we can also have whole genome sequencing, where we sequence the DNA coming out of the bacterium, and Here you can see that there's a SNP that differs between these three isolates, and we would create some kind of network like this. And this UKHSA handbook, which is only a couple of years old, goes into more detail about this MTB cluster investigation handbook. So I suggest reading this. It's a really good way to get an overview of how it's done within the pipeline of UKHSA here in the UK. So we would call this over here a phylogenetic tree, where the tips are things that we have sampled. So we have strain 1 and strain 90 are more related to each other than they are to the others. And then over here we have what's called a minimum spanning network. Sometimes we call them minimum spanning trees. I'm going to explain how both of these work and the pros and cons of each. So first, let's talk a little bit about phylogenetics terminology so that when you're reading a paper or you're reading someone's report, you can understand what the terms are that they're using. So what is a phylogeny? A phylogeny is just an evolutionary relationship between lineages. It's saying that sample 1 and sample 2 are more related to each other than they are to sample 3. So we're saying something like two bird species are more related to each other than they would be to a dolphin. So that's the evolutionary relationships between lineages. And these lineages could be different genes. So the HOX1 gene in a human and a HOX1 gene in a mouse, are they more related or less related to each other than they are to the HOX1 gene in a snake? There could be individuals. So am I more related to my brother than I am to somebody on the street that I don't know? Who could also be my brother and I just don't know it. Differences between populations. How does the population of MTB within the Gambia relate to the population of MTB within Ethiopia or within here in the UK? Or between species. How is Mycobacterium tuberculosis related to Mycobacterium leprae, Mycobacterium canneti? Which of these are more closely related to each other? And phylogenetics is just the study of these relationships. So this is derived from the Greek meaning tribe origin. Actually, phylogenetics doesn't necessarily mean it has to be done on genetic material. They just come from the same word origin. So it's often depicted as a tree with sample taxa at the tips. So this is a standard looking phylogenetic tree. We've got 6 sampled taxa here. So these are 6 maybe MTB samples that I took from different patients. And this is saying that A and B are more related to each other than they are to C, D, E, or F. E and F are more related to each other than they are to the others. And this is depicted by from the sample taxon, which is sometimes called a terminal node, we have a branch that goes to an internal node. And A and B share an internal node or an estimated ancestor, which they do not share with everybody else, and that means that they're more related to each other. So more close they are in tree, the smaller the evolutionary distance between them. Another thing that we would sometimes refer to is what's called a bifurcating node. This is where we have one internal node with 2 ancestors coming off, which is what we want, or a multifurcating node where we have multiple different ancestors coming off. We know that these 3 are more related to each other than they are to these 3 out here, but we don't know which one branched off earlier. So that's just to explain why sometimes you see these kind of straight line ancestors with multiple ones coming off. And I'll explain that a little bit more later. We also can have a terminology referred to as an in-group and an out-group. The in-group is the ones that I care about. That's what I'm studying. That's what I've sampled. That's what I'm trying to investigate. That's maybe my outbreak cluster. That might be my species of interest or something. But we don't know where the root of the tree is. This is the root back here when we're just looking at our in group. We need something, we need to often add in a sample, which is, we definitely know is not related to our group of interest. So an example I'll give you from MTB is we had done this study in Rwanda and Uganda, and we had found a new lineage of MTB, which we call lineage 8. And we needed to know where in the tree it sat. So Mycobacterium tuberculosis has 10 human-associated lineages and 4 animal-associated lineages. We wanted to know, did it sit within a lot of the human lineages, or did it sit within the animal lineages, or where different? So the writing might be a little bit small on this, but you can see based on the coloring here, we have multiple different lineages. And we didn't know, does it sit near the root? Does it sit within one of these? So you need to pick an outgroup And in this case, we picked Mycobacterium kineti, and we know Mycobacterium kineti, the genome from this is going to sit outside of all of the Mycobacterium tuberculosis complex. So we add that sample in just so we know that the root sits along the branch to that. That's what an outgroup is for. It's not necessarily saying anything else. If you don't care where the root is, you don't have to add in an outgroup, but that's just to give you a little bit of an idea of the terminology of in-group versus out-group. So we've got our data, maybe it's oligotype, maybe it's whole genome sequence, and we have decided that we want to build a phylogenetic tree of that. And then you read all these papers and people are talking about parsimony and distance and MSTs and maximum likelihood and Bayesian and all these different things. And how should you pick which one is the correct one for your data? And that's what I'm going to focus on a little bit more for the rest of the talk. And we'll talk about the most simple one first, which is called parsimony. So when we're creating a phylogenetic tree or a phylogeny, we basically are trying to do what this kind of complicated sentence is saying, which is we're using an optimality criterion to define how we measure the fit of the data to a solution. What this means is I have decided that there is an optimal solution for this, and I'm going to use that one to construct a phylogenetic tree from the data that I have, so that the data that I have fits to that phylogenetic tree as best as possible. And this will become more apparent as we go through the different approaches. As an aside, we also have what's called a tree searching method. I'm not going to get into this because of time. As I said, it took taking a bigger course and put this down, but you may sometimes see tree searching in different ones. And I pointed you to some of my online other resources where I explain this a little bit more later. So the simplest method to define an optimality criterion is called parsimony, and it's also referred to as a non-parametric approach. We cannot add any extra parameters into parsimony to try to modify any of the fit. So the optimality criterion for parsimony is I'm going to put together the samples which have the minimum number of changes needed to get from one to the other. So here we're trying to group together things that have the most characteristics in common. So here's an example from a small oligotype. So for oligotype at position 1, 2, 3, and 4, we would only have 23 of these positions. It's either present with a 1 or it's absent with a 0. And we want to put together the ones that have the most present together. So in this one, you can see I'm going to put sequence 1 and sequence 2 together because they share 1, 2, 3 positions where they both have um, a spacer present. So my optimality criterion is put things together that share the most in common. We next put in sequence 3 because sequence 1 and sequence 2 have 1, 2, 3 in common, and they both have 1, 2 in common with sequence 3, so I add that in next. And then sequence 4 I'm adding in last because it has the least in common with everything else. And it builds a tree that looks like this. So if we start from the base, we would say the base has no spacers present, so let's say it would be all zeros. We add in one spacer to get to sequence 4, so we add that one first to the root. We then add in sequence 3 because that's the next one. We're adding in this spacer. Then we're adding in sequence 2 and sequence 1 to create this final phylogenetic tree here. So parsimony is the simplest way. It has one answer at the end. It is just what has everything in common with each other. So if we were doing it with animals, you might say, oh, which ones have beaks and feathers, and which ones have four legs, which have two. We would take all these characteristics and we would say, well, these share the most. So it has the most parsimonious answer to that. But parsimony comes with a lot of problems, and one of them is what's referred to as convergent evolution. If you have a trait that has been evolved independently in two different lineages, parsimony will put them together because they share that trait. A great example of this is wings in bats and birds. Parsimony would put these together because they both have wings, even though we know they evolved them completely independently of each other, so should be put separately. Echolocation in bats and dolphins. Also, we know that that's evolved completely separately from each other, but you may put them together because they both have this ability to do echolocation. So In Spoligotype, a MirUVNTR has a huge convergent evolution problem because Spoligotypes don't just evolve in a stepwise position, position one and then position two, then position three. Two completely independent isolates could have a spacer present in position four completely independently of each other. So in this phylogenetic tree here, you can see that parsimony is used based on Spoligotype patterns, and the coloring and the isolates down here are supposed to indicate Spoligotype groups. And you can see that if you would just follow the T here, we can see that it's here, it's here, it's mixed in here with the lamb, it's also mixed in here with some orphan ones, it's also down here mixed in with Cas, and it can be mixed in over here with the X lineage. So Spoligotype and MirUVNTR really suffer from convergent evolution, which is one of the reasons we don't really use them for phylogenetics or for transmission tracking anymore, because you cannot always tell from a Spoligotype or MirUVNTR pattern if things are truly related or whether convergent evolution just brought them together. So we really want to focus more on whole genome sequencing for anything that we're doing. This is true, I'd say, of almost every pathogen now. If you can afford it, it's a great way to do it. Spoligotype, very, very strong for telling you what lineage your sample is in. MirUVNTR is less susceptible to convergent evolution. So it can be good for kind of large scale survey tracking. And this used to be done like when you want to know if there's a strain across the entirety of the EU or something like that. But we don't really use it for recent transmission anymore because it doesn't have the resolution, and convergent evolution is a big problem. Genomes can also have a convergent evolution problem, and this is very specifically, a great example of this is drug resistance. So here we have a phylogenetic tree. It's exactly the same as the phylogenetic trees that we had before, but it's been curled back on itself just to save space on the page. Good for large trees when you're putting them into publications. It's referred to as a Hillis plot after David Hillis. So you can see that we have a load of isolates here that are related to each other within lineage 4, some here from lineage 1, some from lineage 3, and some from lineage 2. And on the outside here, this inner circle here is denoting when an isolate has one of these CatG mutations. And you can see that it's spread all through the tree. It's not CatG mutations, which are related to isoniazid resistance, didn't just evolve once and then spread throughout the population. They evolve independently in multiple different ones. So if you just rely on those to build parsimony trees, it can really impact and bring together isolates incorrectly. Now, that effect, that convergent evolution effect, is often drowned out because we have so many SNPs across the entire genome. So between a lineage 4 isolate and a lineage 2 isolate, there's going to be 1,000 other SNPs that are going to drive the parsimony to separate them out. But it's something to be aware of if you're working on very, very closely related isolates and they have drug resistance patterns in there, you may want to remove them, or if you're doing parsimony. Although generally I don't really recommend parsimony anymore. It does not really any advantages over the other methods that we'll talk about. So we'll move into distance-based methods now. These are a little bit more common. You may have heard of terms like neighbor joining or UPGMA. So as I said, parsimony is only useful when there's very little differences between isolates and there's not no convergent evolution that's going on, and you've captured all the differences that are there. So you could use it for a local transmission cluster, maybe less than 5 SNPs, which are separated by only a couple of SNPs, as I said. But a distance approach is almost always going to be better than a parsimony approach for the kind of data we're working with. Parsimony is referred to as non-parametric. All changes are weighted the same and are as likely as each other. And I'll talk a little bit in a minute about why that may not be true. Parsimony also has big difficulties with rapidly evolving lineages. Not so true with MTB, but really, if you're working on viruses or you're working on rapidly evolving bacterial pathogens, maybe like E. coli or something like that, parsimony really has problems with that because it can't handle huge amount of changes between samples. Distance-based metrics handle this rapid evolution a little bit better. How distance approaches work is almost the inverse of parsimony. You get the distance between two isolates. So if we go back here, The distance between sample 1 and sample 2 is 1 because there's one difference between them. The difference between sample 1 and sample 3 is going to be 2 because there's two differences, the 1, 1 here and the 0, 0 here. So we're getting a distance as opposed to things that they have in common. We're separating them by things they don't share. And then the sequence with the shortest distances are most related, and we put them together into a tree using what's called a UPGMA approach, or maybe a neighbor joining. You may see BioNJ sometimes, especially if you're working with a program like Mega. Distance-based methods are also what's called semi-parametric. We can weight these differences by saying, oh, a SNP, a change from a G to an A may have a different weight than a change from a G to a C or something like that. So UPGMA essentially is taking, let's say we have these sequences like this, and we would build a distance matrix. So here, I'm counting up the number of differences between two isolates, and I'm dividing it by the total number of sites that are there. So between the S1 and S2, the T is the same, the T is the same, the C is the same. This has an A, and this is a G, so one difference, the G is the same. So 1 out of 5, which is 20%, 0.2. So then between sequence, let's say 3 and sequence 4, we see the same. The A to the T is different. So we have one difference that's 20%. And I fill in an entire matrix like this of the differences between these isolates. Note that we're counting the differences, not the ones in common, and it's differences divided by the total sites. I'm not going to go through step by step. That's going to take up too much time. I put them here. The slides will be shared. They're also up on my website, which I will link to in one of the later slides for you to go through. But essentially, this is the steps that UPGMA goes through to do it. You don't have to do this by hand. That's what programs like Mega are for if you want to be building these kind of distance matrices yourself. But it essentially does some kind of calculations that are there, and it recalculates based on which ones you put together. So you can see that sequence 1 and sequence 2 are coming together because they have the shortest distance. Sequence 3 and sequence 4 are coming together because they also have a very short distance. And sequence 5 is very different from everything, so it sits at the end, is essentially how that works. Now, when I say that we need to weight the differences between them, it's because in evolution, not all nucleotide changes are exactly the same. We have a couple of different things. So actually, I'll come back to this first point in a minute. But we know that transitions and transversions have different rates. So a change from a C to a G will be different from a C to a T and from a C to an A. And that's just because of physical properties of nucleotides, because of different things like that. So we may want to say that we put more weight on a change from maybe a C to an A than we do from a change from a C to a T or a C to a G or something like that. We also may say that in certain positions in the genome, you're more likely to see a change compared to others. So we know that we have hotspots of hypermutability, where we're more likely to see mutations maybe in certain regions of a genome compared to other. And we might want to say that those changes in those positions have a higher weight or a lower weight when we're trying to do these approaches. So it's important to try to to be able to modify the distances between isolates based on this kind of information, and that's called a model of evolution. I'm not going to go, as I said, unfortunately, into the models of evolution in detail. That would be a little bit more maths than we may want to get into today, and has a lot more technical aspects on it. But here I link to my GitHub course. If you just search my name, Conor Meehan, and pathogen data course, you'll find it. And in there, I've got 3-hour course on this phylogenetics. I've got a whole other one on Bayesian and phylodynamics. I've got an optional lecture there also on how models of evolution work. So if you really care about that, dive into them. You can always email me with any questions. But one of the reasons that distance matrices approaches fall down is they still have a problem with convergent evolution, but they also have a problem with what we call unobserved back mutations. So this means that if you're looking at isolates over a long period of time, let's say I have a Mycobacterium tuberculosis sample from lineage 1 and a sample from lineage 5. These diverged probably thousands, tens of thousand years ago. And during that period of time, you could have had what's called an unobserved back mutation. That is, that a mutation went from an A to a G and then went back to an A in that same lineage down to what we observe today. And Distance matrices cannot take that into account, and neither can parsimony. They just see what's in front of them. They'll just see that final A. So actually, you had two mutations where it'll say there are no mutations. So it underestimates the distances. So again, long periods of time, distance approaches have real problems with that. I'm going to do a quick aside on what's called a minimum spanning tree, because you see minimum spanning trees popping up in almost all public health epidemiology papers now. And I wanted to have an aside to kind of outline to you the pitfalls of these approaches and when they are appropriate. So what I'm mostly talking about is building phylogenetic trees. We've talked about parsimony approaches and I've talked about distance approaches for building those trees, but we also have what's called minimum spanning tree uh, approaches, which are different from phylogenetic trees. Not too different, not so much that you should be hung up on the details, but they won't look like phylogenetic trees where you've got this kind of tree-like structure like this. They'll look more like these networks. So even though they're called minimum spanning trees, they're actually networks. So for a distance matrix like we had a second ago, and with a distance-based metric method like neighbor joining or something like that, we're trying to build a network that connects all the nodes with a minimum total distance. So it connects nodes together where it tries to have the total distance from any one point to another point across the whole network to be minimized. That's why it's called a minimum spanning tree. And the difference here is that sample taxa, so if we go back here, all of our sample taxa sit on the outside here. It assumes that these were all maybe infected by an unknown person, which are represented by these internal nodes. Minimum spanning trees are really useful because the samples that you take can also be nodes in the network. So it means that if you have complete sampling across your population, it'll allow you to see maybe who was the ancestor of who else, and it can really help with those kind of ones. So they do have a use, and they work very, very well when you have some SNPs, but not too many SNPs. So in this case, which is coming from this TB paper, they built a phylogenetic tree, they got their transmission clusters, and then within those transmission clusters, they built minimum spanning trees. Low chance of there being back mutations, especially in TB with a low mutation rate, low chance of convergent evolution issues as well. So we uh so in this example here, you can see that they're estimating that this is most likely the ancestor of this little cluster, and most likely this is the first secondary case, which then led on to the first one that they diagnosed, then another case, and then another case. So they're able to reconstruct a little bit who the potential ancestor is and which of the cases came first in this kind of order. And in this case here, they've got four isolates, and they're saying we don't know who the ancestor is, but it's probably none of the ones that we um, sorry, actually here they're saying that the first diagnosed case and is the most likely the start of this transmission cluster as well. And then over here, there's a lot of connections between them. They don't know which ones are the start or the end. They suggest it's potentially this one, but that there are a lot of connections between them as well. So multiple spanning trees are very useful for looking a little bit at the order of infections potentially, not always, but it can help with that. Now, MSTs have a lot of pitfalls, and one is that the selection of the minimum spanning tree is arbitrary. Often, when you only have a couple of SNPs that are different between isolates, multiple different network interactions could explain exactly the same data, and MSTs won't always tell you which one, it'll just give you a result. It doesn't always give you the statistical support to tell you which ones of these is true. So from this paper, they had done minimum spanning trees of Spoligo type data, and they applied different MST approaches. So this one here is, I think, taking the one with the most agreements. This one is taking the one with the fewest changes. And if we just track sample 7, so sample 7 here is connected to sample 22, coming from sample 4. And we want to know its relationship to sample 6, which sits all the way up here. So in one method, it separates sample 6 and 7 quite a lot. In the other method, it has them right beside each other with a 53% support on that. So minimum spanning trees, especially with complex data, can be really, really difficult to interpret and can give you misleading results. So it's good to keep them to really, really small transmission clusters where there's very few SNPs and no convergent evolution. And there is convergent evolution in small oligotype patterns, so again, not so great for them. So just a quick aside on MSTs. Useful in a lot of situations, but make sure that you're being very aware that it can lead you down the wrong path. The issues with distance-based metrics, then we still have a convergent evolution problem, just like we saw in parsimony. It decreases the distance artificially because it thinks that drug-resistant patterns are coming together and such. That's what's called a long branch attraction problem. This is where two isolates are so different from everything else in your sample that they'll sit together in the tree, but they're not actually related to each other. They're just not related to everything else. So that's kind of a very quick description of long branch attraction. The models of evolution are very simple. It's able to do those transitions versus transversions and things like that, but it can't do very complex models, what we call site heterogeneity. You might see in a paper that they said, we built this tree with a gamma distribution for site heterogeneity. So you can't do that with something like a distance approach. And it starts from a single distance matrix and it goes to a single solution. So if there's any kind of difference in the way that you calculated that distance matrix, there's no exploration of that inside of it. So this leads me on to very briefly talking about parametric methods. So parametric methods are where in the first two examples, parsimony and distance, it takes two sequences and either calculates the distance or something between them, and it does that to create an independent a big matrix. Parametric methods actually take the sequence alignment and it goes column by column and it calculates how much support that individual column has for the tree that we're building. And I'll show you this in a second. And there's two main parametric methods of phylogenetic inference, maximum likelihood and Bayesian analysis. I won't talk too much about Bayesian analysis. There is going to be a future webinar from the PHA4GE network on phylodynamics, which is all based on Bayesian, so I will leave it to that. But I put some slides in here to explain the basics if you really want to get into it. They're called parametric methods because they both require an explicit model of evolution. You have to give it parameters. So parametric, you have to explicitly tell it the parameters. And they can incorporate really complex models of evolution, such as what's called the general time reversible model, GTR, and site heterogeneity. So maximum likelihood. I'm going to try to explain maximum likelihood in about 3 minutes. So this is really just a stop gap on it, but it'll give you hopefully some of the basics to be able to go out and explore it yourself if you care about how it works. Focus today is more about why we would use one method over another. How maximum likelihood works is instead of taking a distance matrix and then proposing a tree from that, you actually propose a tree and then you see if that tree fits to the data that you have. So in this example, the computer program has proposed that sequence 1 and sequence 2 sit together, sequence 3 and sequence 4 sit together. They all share a common ancestor, and sequence 5 sits to the outside. And then maximum likelihood goes column by column, and it says, do you support the tree that I have proposed here? So it's like going into a meeting and someone saying, I have a proposal for how this meeting should run. And then everyone votes and says, I agree with that proposal. And if nobody agrees, they may go away, come up with a new proposal, and come back to you. So that's kind of how maximum likelihood is working. So here it's saying we have a T for sequence 1, 2, 3, and 4, and an A for sequence 5. So this would support that sequence 1, 2, 3, and 4 are more related to each other than they are to sequence 5. So we have a likelihood, let's say, of 100. It doesn't matter about the numbers. Column 2 is putting sequence 1 and 2 together. It's putting sequence 3 and 4 together, and it's putting sequence 5 separately because these share a T, these share a G, and this shares an A. This has really high likelihood or high support for this tree. It does that across the entirety of the length of the alignment, and then it adds up that likelihood to call it the total likelihood. And then it proposes a new tree. Maybe it moves sequence 5 up to being here beside sequence 2 and redoes that. And it says, is this likelihood more than the likelihood I proposed from the last tree? If it is, then this becomes my new maximum likelihood tree. And it continues to search through what's called tree space until it comes up with the tree that has the maximum likelihood related to the alignment that it's given. It's a very quick explanation of it. But the difference you need to know is that it's going column by column and checking the support of that column for the tree that you've proposed. And this really doesn't have as much of a problem with convergent evolution It has a lot less of a problem with long branches and all these different ones. So it is the preferred way to build a phylogenetic tree for really anything these days over distance or parsimony approaches. And an additional step we can do when we're looking at maximum likelihood is what's called bootstrapping. And you might see this again in papers where someone says, we ran bootstraps and I've got a bootstrap support of 100% on this branch or something. What bootstrapping does is that it's a test of reliability for an inferred tree. How you do it is you take your original alignment, so you can see that this alignment here and this alignment here, we would normally be looking at alignments, let's say for MTB, of 4.4 million bases, right? Because we're going to do the whole genome. And we may just do it on SNPs, and then we're maybe looking at 1,000 or 2,000 SNPs. More sites, the better. We want to have as much information to support the tree as possible and as much variation between our isolates as possible so that we can try to figure out which ones are more related to each other. How bootstrapping works is it says, okay, what if only one of your sites really is driving this tree and the rest of them is not driving it at all? So what it does is it randomly samples from your alignment and builds a new alignment. So here you can see it sampled column 5, then it sampled column 4, then column 3, then column 4 again, and then column 3 again. And it builds a new alignment of the same length, and it finds the maximum likelihood tree of this alignment. And then it compares this tree to my original tree, and it says, do I support the branches? So it's doing this maybe 1,000 times. And it says, of these 1,000 trees that I built where I resampled, how often did sample 1 and sample 2 come together? If they always came together, then I give it 100% bootstrap support, 1,000 out of 1,000. If they came together 500 times, then I have a 50% bootstrap support. So it's saying that there's not a very strong support for this clade if I only have a bootstrap support of 50. It means that it was very tenuously saying that these two came together, that I'm not actually really well supported in my assumption of this. So that's how bootstrapping is working. It's giving you a reliability metric of how often your two isolates came together in the phylogenetic tree. So you can say, I'm really confident that these two are related, and I'm less confident about these others. And if we go all the way back to this MST, that's what these numbers are here. So it's saying that 7 and 6 came together 53% of the time here, but 6 and 7 basically didn't come together at all over here. 7 was more related. But 7 and 4 only came together 8% here, so really low support. It's important to always ensure that your work is being supported. So in MTB, maximum likelihood is the recommended approach for building very large data sets. How we would normally go about it is we're going to take all of our data that we're working on and build an maximum likelihood phylogeny from that. And then from that, we're going to pick subclusters, which might be our transmission clusters. Maybe we're doing a 5 SNP cutoff or something like that. And within them, we could do minimum spanning trees, MSTs, to then try to figure out order of relationships or whether it's kind of more of a network of interactions, more so than just this binary structure. We would often exclude drug resistance positions when we're doing these SNP cutoffs. And when we have these trees, we can do loads of great things. So this is one of my favorite papers for showing it from 2015. This is from KwaZulu-Natal in South Africa. There's a large multi-drug resistant ongoing clone that spreads in that area. And you can see that they have samples kind of going back into the 90s and stuff. And they've basically shown that every time a new drug was found, so you can see streptomycin was introduced here in the 50s, and this strain had already gained that a mutation to that by the 1960s here. This is more of a phylogenetic approach here, but what we're doing is we're lining up our drug resistance patterns here and finding where are the entry points? When do these drug resistances appear in this clone? And maximum likelihood trees can be lined up against other information, maybe region to do phylogeographic analyses or something like that. Now, actually, it's much better to do these in what's called a phylogenomic approach, which will be covered in that webinar in a couple of months. So finally, before we finish up, I want to quickly introduce the concept of Bayesian approaches or phylodynamics, which I said is going to be covered more intensely elsewhere, but it is really for public health, the way that we're going, if you can do it. But Bayesian is a little bit of an art form. It's not so straightforward. Maximum likelihood, get your samples, build your alignment or some kind of SNP alignment, put it into something like Raxamel. I've listed them here. Raxamel, IQ-Tree. These are our two main ones for building trees. It should be actually Raxamel NG is the name of the program now. And it'll give you a tree at the end, and it's pretty straightforward to do it, and it doesn't take too long. Bayesian, you want to work with somebody who knows how to do this, but it can be really powerful at the end. So Bayesian allows you to build time trees where the length of the branch correspond to certain lengths of time. And you can see that in maybe this tree here, even though I put it in the maximum likelihood bit, this is actually a time tree, where the length of the branch is related to time. So this sample is taken in around 2010, and it's estimating all the way back here. We also can add in a lot of other information. So we can add in the date of isolation for all of our samples or the location of the sample or something like that. And we incorporate these all into more of a large data model as opposed to just data matrices or alignments. And then it gives what's called a posterior probability of the hypothesis is true given the observed data and priors. I'm not going to explain too much how that is without getting too much into the details, as it's going to be covered in a future lecture, webinar. But some example programs, if you're seeing these in papers, BEAST and BEAST2 would be the most common of these, but also PhyloBayes and MrBayes and RevBayes. Again, the
Hi,
slides are going to be
Conor.
shared. This explains how Bayesian analysis works. I won't go through it now, so we leave time for questions, but it's just so you can see that it's this kind of like circular approach in what's called a Monte Carlo Markov chain, where you're accepting or rejecting trees, and then you're changing the lengths of the branches and you're changing the positions of isolates. But you can also change things like where did this isolate occur, and you try to estimate extra things. And this all fits under the umbrella of phylogenetics, where we're taking information like, is this a travel related or an imported case? Is this a nosocomial or a community infection? Is this a phenotype, maybe drug resistance or virulence? And we're adding all that into a model along with the phylogenetic information to come up with things like what's the first detected isolate? Where did our isolate break out? What kind of virulence are we seeing? What kind of duration infection? Lots of these things. And this Nature Reviews papers by Atwood et al. Really sums up phylogenomics really well. But from our point of view, where phylogenomics can be really useful is transmission clustering, telling us the order of who infected whom, and really interestingly, do we have any unsampled cases? So in this program, Transphylo, and it's also kind of outlined in this paper down here, we can see we sampled maybe sample one and sample two and maybe sample four, but we missed maybe this sample three, maybe we missed the sample seven. So they also can give us an idea of how good our sampling is throughout the work that we're working with. So I think I've come pretty well on time now. So to wrap that up, that is a very brief walk through phylogenetics. If you are interested in this, as I said, you can go also to my website or email me. They're on the front of the slides, or look up Conor Meehan, NTU. And I'm happy to point you to more courses if you're really interested in the nitty-gritty of how we do these ones. But what I want you to take away today is understanding the four main ways we can do this: parsimony, distance, maximum likelihood, and Bayesian. And we're opting for maximum likelihood in almost all situations now because of the problems with distance and parsimony. And then Bayesian is a little bit more of an art form. It is the fantastic thing for public health, but it can be limiting and very difficult to use. So good to work with someone who's an expert. We know how to do these non-parametric or semi-parametric approaches and MST, and we're knowing with these minimum spanning trees it can be really useful for in some situations, but be very careful that they can lead you down the wrong path. But really what you need to always think about is what is my data and what method is related to that. And that comes back to you defining your question. And I'm happy to take questions on any of that now as well. So thanks for listening.
Hi, Conor. Thank you very much for the presentation. I'd just like to open the floor again and remind everyone that they can post their questions in the Q&A section or within the chat. In the meantime, I do have a question so long, Conor.
Great.
So somebody asked, how were the greater than 5 but less than 12 SNP cutoffs for the cluster classification determined?
Yeah, so actually in MTB they were determined back in about 2013 by this guy Tim Walker and team, and how they did it was actually from an Oxfordshire in the UK outbreak. They had they had epidemiological data relating to the cases that they saw. And they found that people where there was a known link between them, so we could tell maybe brother and sister, or they went to the same pub or the same groceries or something, they could tell they tended to have less than 5 SNP difference between the genomes of their MTB. And anyone where there was no known link, they often had more than 12 isolate SNPs. Now, this was done in a low incidence setting. It was done only with lineage 4, so it has not really been tested fully outside. And some preliminary stuff that my group are working on is actually trying to contest that. So it works generally okay, but take them with a pinch of salt. But for doing those MSTs, at least it gives you something, a smaller number to look at to try to estimate some more recent transmission stuff.
Excellent. And then just another question. This is sort of a bit of a three-parter. The first section is, what is the optimal MCMC for bacteria? The second part is, would you calculate it? And then the third part of the question is, does it relate to sample size?
Right. It doesn't necessarily relate to sample size, so I will start with that. It relates to how long the Bayesian takes to get to the answer, which is not really a very easy way to explain it. But how Bayesian works, I will very quickly talk about it, is that it basically proposes a tree and then it and the same way that maximum likelihood, it checks it against its whole model and says, what is the posterior probability instead of maximum likelihood of this tree being true? And it keeps on doing that until it converges down until every time it proposes a change, it's worse. So you need to get to this convergent point where every time it tries something new, it never improves upon itself. So in Bayesian MCMC, you're looking for a convergence point. And what is that convergence point? Fantastic question. There is not really any way to know it. So when I do it in MTB, we normally have to do about 4 or 5 million length because we have very few SNPs. So we need to run it for very long periods of time. If you were doing it on something like HIV, probably like 100,000 would be enough because there's so many SNP differences between them, it can converge on that answer really, really quick. So this is why Bayesian is a lot of like you run it and then you check it and you see if it's good and then you run it again and then you check it and it's much more of an art form in that way. So yeah, it's all about this convergent testing and going through to get to it, but it's definitely a little bit more of an art form.
Thank you very much. And then just a question from my end. So do you foresee a future where phylogenetic inference is actually incorporated or incorporates larger genomic changes, something like structural variants or insertions and deletions or mobile elements alongside these point mutations?
Right. So there's maybe two parts to that. In theory, a phylogenetic method will take into account indels. So it doesn't do very well, and there's definitely people working on that. So instead of it being a change from an A to a C or an A to a G, it has a change from an A to a gap.
Mm.
And a lot of the programs don't do fantastic with that, but they will use them. So you should include the indels. SNP alignments are actually not preferred for phylogenetic methods. You should be giving it the whole genome alignment with the indels as well. So that's one aspect. Mobile rearrangements is moving from phylogenetic trees into phylogenetic networks. And something like splits tree can do that, but nothing really has done better than splits tree in the, I want to say 15 years since it was invented. But I think we're moving that way. Because at the moment, if you're working on something like an E. coli, only about 40% of that information is going to be vertically inherited through a tree-like structure. We want to be bringing in that whole pan genome and how are things being related, especially yeah, I care that these are related down through their cells, but these are sharing the plasmids, so actually that's more interesting. So I think we're going to see these phylogenetic networks becoming up more and more as time goes on, for sure. So definitely watch, not this space, I don't make them, but watch this space.
Yeah. Then just another question in chat from Noor. If the methods used for whole genome sequencing perform poorly for certain regions, for example, repetitive regions, Are these regions then excluded from consideration when looking for differences in SNPs? And then the second part of the question is, is it always, or do we always not consider SNPs associated with resistance?
Right. So MTB is a little bit of a specific case because it has this what we call masking of repeat regions. This is when you are mapping the reads down. We don't map them into regions which are repeated around the genome. This is in theory because it creates false positives in the mapping. So it's not really about the phylogenetics. Yes, so we do normally mask out those regions in MTB, but actually it's again never really been truly tested about whether you do need to do that. That is something my group is currently working on, and it seems it depends as well. So we often mask them out because you don't know if it's a false positive or a false negative. But that doesn't mean that that is definitely the way you should go about. We do often, for the SNP cutoffs, we don't consider drug resistant ones, so we do exclude them. But once you're within those MSTs, you may want to include them for something like MTB because drug resistance at such a short distance tends to only evolve once. Convergent evolution is unlikely with MTB at such a short distance, so you may want to include them to separate things out within small transmission clusters.
No. I'm just going to then one more question. Um, is there a significant, or does there have to be a significant time difference between samples? For example, like with a clone when performing Bayesian analysis. And what is the smallest sample size on which you have performed this type of analysis?
So I've done it on within transmission clusters. We did it over for MTB, we did it over a 20-year period in Rwanda. It depends on the question that you're trying to answer. So if you want to say what is the origin of all of MTB and when did that occur, you can't really get that from 3 years worth of data because it needs to estimate so far back in the time. Good thing about Bayesian is that it'll give you confidence intervals in any estimate that it gives you. So those confidence intervals would be huge. It's more about how many SNP differences there are. If everything is identical to each other, then you just can't build a tree. So for Bayesian, you normally want to have more SNPs if possible, or what's called phylogenetically informative sites. So you need to kind of have enough differences that the tree can make decisions between, or it'll just never converge. That's why phylogenetic approaches work much better for viruses, which mutate much quicker. So an example we'll give is that like HIV will mutate within a person, have more changes than like all of influenza ever. And then all of influenza has much more than you will ever see in the entirety of TB. So it's more about how quick that mutations are occurring compared to the amount of time that you're sampling for.
Yeah. Okay, one last question, just before we close off. I've got a question from Tracy. If you use a hybrid approach for like Illumina and ONT, can you then include the repetitive regions?
Yes, repetitive region mapping is only because short reads are the difficulty. So as we move into long reads and we are able to cover the entirety of the repetitive region, then we don't have this false positive, false negative problem with that comes from the mapping of short reads. So so as we move towards nanopore and closed genomes and we move away from read mapping, a lot of those repetitive region issues with MTB will go away. And I guess watch this space because we will actually be coming out with a paper on that very soon.
No, excellent. All right, then I will be closing today's session. So thank you all for joining and for everyone's thoughtful engagement. It was a pleasure to moderate everyone. I hope the session shed light on the complexities of phylogenetic method selections for MTB. And a special thank you, Connor, for your valuable insights on this matter.
No problem.
And I would just again like to remind everyone about the PHA Phylogenetics Working Group and any of the upcoming webinars that we are planning on having. Sorry. Thank you again, and we hope to see you in future sessions.